Thank you everyone for attending this webinar. In this session, I will talk about the generation and application of anti-idiotype antibody in biologics development. During its presentation, I will first give an overview of application of anti-idiotype antibody during the preclinical and clinical study. Then I will go over the general strategies of anti-id generation and kit development. Lastly, we will discuss some of the technical challenges in details during the process and provide solutions. The typical application for anti-id antibodies are PK&ADA study. So what is the PK&ADA? To put in a simple word, PK is to measure the antibody drug levels in testing samples, while ADA is to measure the anti-drug antibody and the neutralization antibody in testing samples. How is PK achieved? Mass spec is widely used for the measurement of small molecule drugs. However, antibody drug is totally a different story. Human serum contains 5 to 10 mg per ml of IgG1, while the regular levels of antibody drug require sensitivity of nanogram per ml range. Thus, this assay must be able to pick up the signal of antibody drug among a million-fold excess of signal. Typically, the immune-based ligand-binding assays are applied, such as ELLISA through the drug target or anti-id. An anti-id is the preferred one due to the better stability and the reproducibility. While for an immunogenicity study, anti-id antibody will serve as positive control for measuring the anti-drug antibodies and the neutralization antibody levels for evaluation of the ADA effect on drug efficacy. Here is the roadmap recommended by FDA regarding the development of biologics. PK and ADA analysis are considered as the most crucial part throughout the whole preclinical and clinical study. So what is anti-idiotype antibody? It's an antibody that is able to specially recognize antibody drug through the available region. This specificity makes it perfect tools to measure the antibody drug, and its resemblance to anti-drug antibody makes a good reference for measuring in the immunogenicity study. Based on the recognition's interface on the drug, anti-id can be further categorized as antigen blocking, non-blocking, and complex specific. When the anti-id binds to the drug through the CDR region, it will compete against the drug target and only binds to the free drug. If an anti-ID binds to the drug through the neighborhood regions of CDR, within the framework of a variable region, it won't compete against the drug target, and as a result, it can recognize both bound drug and free drug. If an anti-ID only binds to the drug target complex, it will only recognize the bound drug. Generally speaking, the preclinical development of therapeutic biologics include roughly 12 to 15 months of the lead discovery and 15 to 18 months on the CMC process development. So it's better to give 6 to 9 months ahead of time of the generation of anti-ID for PK and ADA studies, which means you may need to consider starting this work as early as the stable cell line generation stage. Next, I will go over the general strategies for raising the anti-id antibody. Currently, the major platform for antibody generation includes hybridoma, phage display, single B-cell cloning. Hybridoma is probably the most popular method due to its cost efficiency, flexibility. For monoclonal antibody generation, we recommend to start with animal immunization, followed by fusion, screening, and subcloning. The subcloned hybridoma will be scaled up for antibody production and purification. We can also use phage display method for the development of anti-ID antibodies. The synthetic library approach will shorten the timeline to 2 to 3 months. For polyclonal antibody, the rabbit and goat are preferred for immunization, followed by antigen affinity purification and cross-absorption with isotype control. So with all those options, we recommend to choose a suitable package based on the final application. Mouse and rabbit monoclonal anti-AID are suitable for preclinical PK studies, while the rabbit polyclonal antibody is only acceptable for early-stage PK and ideal for an ADA study. The detection sensitivity of the rabbit monoclonal antibody is generally better than mouse, but the mouse version is typically enough for the purpose since the dose of the antibody drug is usually higher than the small molecule drugs. Over the last decades, more and more non-traditional formats of antibody drugs appears on the market, such as ADC and bispecifics, and the strategies of raising the anti-AID antibodies against the newcomers are also different. However, the key is still to develop anti-AID against the vulnerable region. Taking ADC as an example, if you want to detect intact drug, recommended to use anti-AID against the small molecule pairs with anti-AID against the antibody carriers. But if you want to measure the level of an antibody, no matter whether it's intact or not, anti-AID against the antibody carriers will do the job. If you are only measuring the level of small molecules, LC-MS is a better way. Similar, for the case of bispecific antibody, if you want to develop an antibody to detect the intact molecules, we recommend to use anti-AID against one of the monospecific unit pairs with another anti-AID specific against the other monospecific unit. But if you just want to know either one of the monospecific unit, anti-AID against the one unit will be enough. After obtaining the good anti-AID monoclonal antibodies, assay development with suitable format will directly affect the outcomes of the PK studies. So in the PK studies, a direct antigen capture method does not require the development of anti-AID antibody. However, the method has poor sensitivity and accuracy. And the anti-AID capture method and the sandwich ELLISA method are probably the most popular formats commonly used in the assay. By capturing the antibody drugs with anti-ID and detection through the anti-human IgG antibody, the capture ELLISA is able to achieve the pretty good sensitivity within a relatively low cost and timeline. However, using the anti-human IgG antibodies commonly leads to the issues like high background and less than ideal specificity. On the contrast, sandwich ELLISA with pairs of anti-ID can be associated with the drugs simultaneously. It's able to achieve the better specificity, sensitivity, but may come with a higher cost. For ADA study, it's pretty simple. The bridging ELLISA format using the antibody as a coating and detection reagents is commonly used. For the last part, we will discuss some of the technical challenges during the generation of anti-AID antibodies. First of all, the variable regions may not be immunogenic, and the constant regions take a large part of the antibody drug. As a result, only a small portion of the antibodies are actually an anti-idiotype. For the immunogenicity issues, we may conjugate carrier proteins such as KLH or BSA to enhance the immune response for the smaller drugs like SLV or nanobodies. Alternatively, we can use a Fab fragment as immunogen to reduce the Fc-specific antibodies. In addition, different adjuvants, animal strains, and immunization schedules can also be included for the maximal success rate. Regarding the issue of low percentage of anti-AID during monoclonal campaigns, we use the high-throughput screening to increase the scales and use higher coatings concentrations of isotype controls to exclude the Fc-specific antibodies. Typically, at least 2 runs of ELLISA-based screening will be carried out to select out those positive against the target drugs, but negative to the isotype control. For polyclonal antibody purification, we also need a better solution to deal with the issue of low percentage of anti-ID in the serum. Typically, we first purify the serum with drug IgG through the affinity column, and then do a cross-absorption with total human IgG. At this point, we will test the titers of purified antibodies against the drug IgG and isotype control to determine whether needs further runs of cross-absorption with isotype IgG. If your drug comes with IgG2 or IgG4 isotype, you probably need to consider doing these extra steps. In addition, we also recommend to optimize the selection of anti-AID based on the requirement of final applications through a bunch of assays including Affinity Ranking, Epitope binding, pairing, and Antigen Blocking and competitive assays. To obtain a high-affinity anti-AID monoclonal antibodies, Affinity Ranking can be achieved by ELLISA-based method, which is relatively cheap, or through an SPR-based off-rate ranking. In order to obtain the different types of anti-AID antibodies, we can differentiate that by competing or blocking ELLISA. In this assay, the drug IgG will be coated on the plate, followed by incubation of anti-AID antibodies and the drug's target antigen. The goal is to determine whether the anti-AID is able to inhibit the association of the drug and its target antigen. In order to screening for the antibody pairings for PK study, we will first perform epitope binding to identify the anti-AID monoclonal antibodies with different epitopes. Epitope binding can be achieved by ELLISA or Biologics. In the ELLISA format, every 2 different anti-ID monoclonal antibodies are incubated with coated drug for binding competitors. Decreasing signals implies the competitions due to the same or close epitopes. In the Biologics method, drug IgG is first saturated with one of the anti-AID and followed by the addition of a second anti-AID. So higher binding signals suggest a different epitope. Once obtaining the anti-AID with different epitopes, sandwich ELLISA will be performed to identify the antibody pairs. In the sandwich ELLISA, one of the anti-ID is coated on the plate as capture antibody, and the other anti-AID is used as detection antibody. The successful pairing will form a complex with drug IgG in between. Besides the antibody generation part, the assay development also comes with its own challenges. To improve the success rate, it's recommended to perform the pilot studies and initiate sensitivity determination to select out the best pairs of the anti-ID from the downstream assay development. In addition, assay optimization with stringent QC standards, assay validations with actual testing samples will also helpful to minimize the risk. In establishment of the PK studies, we need to determine the sensitivity and detection limits. So how is the sensitivity calculated? First, we need to take an average of the 20 repeat OD values at 0 concentrations and calculate the standard deviation. Then we substitute the mean values plus 2 times of the standard deviation into the standard curve to get the final calculated concentration. In addition to that, the inter and intra-assay variation are also the key parameters in the PK assay development. The intra-assay difference should be controlled at 15%, and the intra-batch difference is controlled at 10%. Besides that, the recovery rate is also critical QC parameters for the kit development. The non-concentrations of standard samples will be tested, and the detect OD is used for calculations of the concentration. The difference between the standard concentration and the calculated concentration is called the recovery rate, and is generally need to be controlled within 80 to 120%. As a critical reagents in the biologic development, anti-idiotype antibody generation and kit development require strict compliance requirements, including data integrity, laboratory recording, instrument certification. Transcript ProBio has an abundant experience on anti-idiotype antibody generation, and also the complete compliance. To summary, anti-idiotype is critical reagents for preclinical and clinical studies for therapeutic biologics, and the time of an anti-idiotype generation needs to be coordinated with CMC stage, and the types of anti-idiotype is determined by the drug targets and the requirement of the sensitivity. Thank you all for your attention, and now we are ready for the question sessions.